Okay, in this video we're going to talk about homeostasis. Now, homeostasis is the maintenance of relatively stable internal conditions despite continuous changes in the environment. So what we find then is that our bodies are exposed to factors that can change the internal state. And what we find then is that our organ systems, all 11 of them, need to work together in order to maintain homeostasis, which is this dynamic state of equilibrium or internal balance. So the reason why it's dynamic is that it's always readjusting as needed. So that if your blood pH becomes too acidic, well, we have organ systems that make the appropriate adjustments. Or if your body temperature gets too low, we have certain organ systems that also make the appropriate adjustments. And that way we can basically help maintain the stable internal environment. And it's actually maintained by contributions of all of your organ systems. So in order to maintain homeostasis, we have to constantly monitor your internal body state. And so it turns out your nervous and endocrine systems, as well as other systems, play a major role in maintaining homeostasis. And they do this by measuring variables. Now, variables are those factors that can change. Things like blood sugar, body temperature, blood volume, blood pH, and stretch of certain organs. These are all variables. Now, homeostatic control of those variables requires three essential components. We need to have a receptor, which actually picks up information or stimuli. We have to have a control center, which is like the processing center of this evaluation. And then an effector, which actually carries out the response. So receptors are the sensors. And then what they do is they monitor the environment, either outside of your body or inside of your body. And these receptors can respond to stimuli. Now, stimuli are things that can cause changes in variables. An example of a stimulus could be like a warmer body temperature. Or another example of a stimulus could be like a more basic blood pH or more sugar content in your blood. Because these are variables that can change, all of those could be stimuli, and we have receptors for those stimuli. So we have specific types of receptors that can respond to those variables. We have receptors for blood sugar, receptors for blood pH, or even receptors for body temperature. And what receptors do is they pick up this information and essentially send that information to control centers. Now, control centers are those which determine the set point, which is the place you want those variables to be at, right? So think of the control center as like the thermostat of this process, where a thermostat contains a set point, which is the temperature at which you want your room to be. However, we have control centers of our body, which also include set points, like for body temperature. And this receives input from your receptors, and it therefore also determines the appropriate response. Receptors might pick up information about a particular body temperature. That receptor sends information to a control center, but if the body temperature is at set point, which is the ideal point, then the control center just won't do anything. It won't enact any changes. However, if your body temperature is too low, it'll determine the appropriate response and then tell the appropriate effector what to do. So effectors receives output from control center, and it provides a means to respond to a stimulus. Now, effectors can respond in a variety of ways, either through negative or positive feedback. If the effector helps to reduce the stimulus, we call that negative feedback. If it enhances the stimulus, we call that positive feedback. Now, negative feedback is the most widely used feedback mechanism in the body because it helps to reduce or shut off the original stimulus. So if the variable changes, like body temperature gets too cold, negative feedback would help to prevent your body temperature from getting colder. So the variable changes in the opposite direction. So therefore, if your body temperature is getting colder, negative feedback would help to warm your body up. And examples of this, again, body temperature, even blood glucose. So for instance, if your blood glucose or blood sugar gets too high, your body can release a hormone, insulin, which helps to reduce your blood sugar and prevents it from getting too high. We call this negative feedback because it helps to reduce or shut off the original stimulus. Now, examples of negative feedback would include things like receptors that can sense blood glucose. Your pancreas in this case is the control center, which can help to secrete insulin when blood glucose levels get too high. And then insulin causes body cells, which would be your effectors, to absorb more glucose, which can therefore decrease your blood glucose levels. So remember, we got those three essential components here. We got receptors that sense increased blood glucose. We got a control center here that actually helps to determine the appropriate response. In this case, is the pancreas. And it secretes a signaling molecule, insulin, which tells the appropriate effector, your body cells, to absorb more glucose in order to decrease your blood glucose. And this is an example of negative feedback. So just to summarize this process, remember, feedback mechanisms require three essential components. We got a receptor, control center, and effector. Receptors are the things that detect changes, and they actually pick up information, which is a stimulus. And then receptors send information to a control center, but they do this through what we call an afferent pathway. An afferent pathway, it means just bringing information towards the control center. And then control centers decide what to do with that information, and then basically can communicate with the appropriate effectors through the efferent pathway. And then the effector carries out the appropriate response to help bring you back to normal balance. So an example of this negative feedback process could be things like body temperature regulation. So let's use the example of your body temperature gets warmer, right? So body temperature rises. We got receptors for this. And then these thermoreceptors for temperature send information towards your brain through an afferent pathway. And then this control center of your brain can actually communicate with the appropriate effector, which is in this case sweat glands. So sweat glands turn on when your brain tells them to. And then those sweat glands help to reduce your body temperature through evaporative cooling to hopefully get you back down towards a normal body temperature. And you can talk about this in the opposite way too. Let's say if your body temperature gets too cold. So your body temperature gets too low. We still have receptors that can pick up temperature information. That information is sent to a control center through an afferent pathway. Control centers decide what to do with information. In this case, it's the thermoregulatory center of your brain. And then here, the control center of your brain can actually communicate with the appropriate skeletal muscle effector through the efferent pathway, causing you to shiver and then warm your body temperature back up to normal balance. So in this example, you can see we have two opposing negative feedback mechanisms that help keep you in internal balance, which is homeostasis. Now for positive feedback, they're much more rare because the responses here exaggerate the original stimulus. So this is where you have a cascade or amplifying effect where feedback causes a variable to continue in the same direction as the initial change. So it usually controls infrequent events, things that don't require continuous adjustment, because if most of your body relied on positive feedback, an example of this would be as if your body became cooler and then you had responses that made you more cold, right? Because you're enhancing the original stimulus, and that would make sense with maintaining internal balance. But sometimes positive feedback makes sense. An example of this could be things like during labor, right? So when the uterus starts to contract, we get the release of oxytocin, which causes stronger and stronger uterine contractions. Or during blood clotting, where platelet plugs stimulate more and more platelets to aggregate and stick, so the existence of a clot causes larger blood clots to grow. And so you can see that here as an example of positive feedback, where if you have a ruptured blood vessel, a break or tear in the vessel wall causes platelets to adhere. They release chemicals that attract more platelets, and then those platelets also release more chemicals that attract more platelets. This is an example of positive feedback, which allows your blood clot to grow larger and eventually plug this ruptured vessel. So in this example, positive feedback makes sense. But remember, positive feedback helps to enhance the original stimulus. So the original stimulus here is platelets aggregating. And so this is positive feedback because these platelets can release more chemicals that attract more platelets. So that's positive feedback because you're basically enhancing the original stimulus.